So first of all, welcome and looking forward. This is very exciting. Universal sequencing is a super low input linked read library prep for accurate long range sequencing with short read, with short input. Okay, let's next slide. We provide innovative low input, which is a very small DNA, super long read DNA sequencing solution. Our Telseq technology simply turns a second generation short read DNA sequencer into third generation long read sequencer. The affordable Telseq library kit is used for microbial genome assembly, metagenomics, de novo assembly, whole genome phasing, and phase whole exome and target sequencing and structural variant identification. We provide the kit, the analysis pipeline, and all you need are the samples and some common lab equipment. We see the growth is tremendous for the NGS market. Looking at 16 billion last year and growing to 32 billion based on BCC research. As you can see here, the US, North America is 46%. When you look at Europe, which represents 22% of the total global market, we see some indication that this will change and is much higher, but we'll keep following up on that. So we're in the right time. A big unmet need for the long read. As you can see, the short read, the second generation dominant with 92%. Illumina controls over 80% of the market, which, this is the market that we're going after with our Telseq product. The long read, the third generation, is far behind. PacBio, for example, is too expensive, and ONT not accurate. And I hear a lot of comments about the support from ONT is degrading, so we get a lot of that as well. Actual need for long read is 30% of the market. It's 7 times more than today's long read market. The main thing is a summary here for what we offer. As you can see in the picture, is a Telseq library combined with the Illumina short read sequencer that gives you the solution that we're looking at. So it's a simple one tube. Andrew will describe to you how easy it is. A fast and easy to use link read library kit. It converts a short read sequencer into a highly accurate long read sequencer and requires very low input, 0.1 to 5 nanogram DNA. The application, as we described earlier, it falls into these categories: de novo, microbial and medium to large genomes, metagenomic analysis, structural variation detection, human whole genome phasing, and phased whole exome and target sequencing.
All right.
And I will over to Andrew now to talk about the technology and background.
All right. So, hi there. I'm Andrew Anfora. I am our FAS manager. So moving forward, I would be a great point of contact for any of your technical questions, or you can reach out to our tech support email as well, which is largely manned by me, but others also see. So I'm going to walk through some of the technology and just how our assay works, and then go into some examples. So we will have time for Q&A at the end. If there are any urgent questions, feel free to speak up. I do have a lot of exciting data to show, so want to try and save questions to the end until they're urgent, unless they're urgent. When we're talking about next generation sequencing, the first 2/3 of this slide should be review. What we see is a diagram depicting a diploid genome where one chromosome is colored, its haplotypes are in blue, and the other chromosome has its haplotypes depicted in a red coloration. When we are talking about short read sequencing, we're primarily talking about Illumina. And we're talking about standard workflows where library fragments will be between 100 and 500 base pairs in length. These are going to be highly accurate reads. However, the short read length does mean that it is nearly impossible to phase haplotypes unless they're within 500 base pairs of one another. It is also quite challenging to perform de novo assemblies using short reads. So moving down the chart, Here's a representation of long reads. And long reads, for the purposes of this talk, are going to be either PacBio or Oxford Nanopore generated data. With the case of PacBio, the customer can choose between higher accuracy with moderate length or lower accuracy with greater length. Oxford Nanopore tends to have lower accuracy than PacBio, just in general. With these long reads, you are able to have a better chance of linking haplotypes and being able to phase the genome. Also, the longer read lengths do allow for more robust de novo assembly than unlinked short reads. Additionally, the cost per base pair acquired tends to be higher for long read platforms than the Illumina platform. So our solution provided by Universal Sequencing is the Telseq assay, which prior to DNA fragmentation will apply a unique a UMI barcode to DNA molecules that allows us to trace back the short read library fragments to a molecule of origin. This lets us link the reads and generate sequencing equivalent molecules of up to 100, over 100 kb in length. Later on in my talk, I will show you some data sets where we have a substantial amount of molecules greater than 100 kb in length that really let us phase large structural variants and perform robust de novo assemblies. And I'd like to go a little more into the molecular detail of how we prepare these linked read libraries. Before I walk through the specifics, I do like to emphasize, as Aaron mentioned earlier, that our assay reagent doesn't require exotic instrumentation. If you have a customer that's already preparing next-gen sequencing libraries in their laboratory, they should have all of the required equipment to prepare our libraries. The library prep takes place in standard PCR tubes. Again, very easy and straightforward. So to walk through our protocol, we'll move from left to right across this diagram. What we first do is we require a high molecular weight DNA to get the best data out of our system. I'll have a slide showing the relationship between DNA length and phase block size later on in my talk. But ideally, you want customers to isolate DNA molecules greater than 40 kb in length. That's really where they'll see the most impressive assemblies with our technology. Not everybody and not every experimental system can achieve 40 kb DNA molecules. As long as you can get molecules greater than 10 kb in length, there will probably be data improvements by using our assay. If somebody cannot isolate DNA greater than 10 kb in length, they might not see much benefit from our technology. So with that high molecular weight DNA, customers will mix a transpososome complex as well as our barcoded TEL beads. The TEL beads have unique identifiers. We use an 18 base pair barcode. So there are approximately 2 billion UMI combinations, so we can generate a quite complex library and are able to handle large genomes quite well due to the very diverse barcode library coded on our beads. So each bead will have a unique 18 base pair barcode. So when the beads, DNA, and first transpososome are mixed together, they will form a complex with the transpososome binding the barcode site as well as randomly binding the long DNA molecules. Our default reaction conditions should have approximately 6 to 10 long DNA molecules binding each bead. We are able to assign molecule of origin when we examine any two library fragments, if they share a UMI and are within 50 kb of one another, we assume they come from the same molecule. If two library fragments with the same UMI barcode are further than 50 kb from one another, we assume separate unique DNA molecules of origin. To this complex, we will tagment with a second transpososome. Then we will break and wash the DNA. And a subset of the long molecule will contain both transpososome priming sites. And we will amplify the clean broken DNA with a PCR reaction, and the resulting library fragments will have the P5 and P7 adapters compatible with Illumina sequencers produced at the end. The whole workflow is quite simple relative to most library preps. The hands-on time, the total time from isolated DNA to sequenceable library is about 3 hours. So once a customer is familiar and comfortable with our library prep, it certainly would be possible to isolate DNA and start sequencing in the same day that you are preparing our libraries. So again, it's a very easy protocol to do. If there are challenges, it's usually organism specific, isolating high molecular weight DNA, and we can provide some assistance with that as well if there are questions. Once you have collected your sequencing data, we have several analysis options, and I want to walk through two of the major options that are depicted in this diagram. So everything that is colored in green on this illustration is software provided by Universal Sequencing free of charge. And so I want to walk through the green path first to describe our analysis. So just like any other project, your sequencing data will come off the sequencer. At that point, customer is required to run Telread, which is our QC pipeline, and also will error correct, apply some error correction to the UMI barcodes. At this point, you will have cleaned up FASTQ files ready for further analysis. Two of our currently released tools are Telseq, which is our de novo assembly pipeline. It performs best for small and mid-sized genomes. So we recommend using it for genomes up to 200 megabase in size. We also have Telsort, which is a phasing pipeline for phasing structural variant analysis. Telseq and Telread are currently available on Illumina's base space, and Telsort will be available either very late this quarter or very early next quarter, sometime in the March-April timeline, I believe. Additionally, all of the software are freely downloadable as either Docker or Singularity images that can be accessed from our website. So that is the analysis options using our tools. Our data type are very similar to the legacy 10x Genomics linked read offering. So we do also provide a conversion tool that converts our 18 base pair indexed libraries to a 16 base pair format compatible with 10x. And that allows people that prefer 10x's software to use our data with Long Ranger or Supernova. And a lot of customers that are looking at larger genomes will use Supernova for the de novo assembly. We're working to improve Telseq for larger genomes, of course, but generally for very large genomes, Supernova currently performs a little better. Not depicted in this illustration is any sort of third option. So if somebody runs, once they run Telread, if they want to do something else with the FASTQ files, they certainly can. They will have a pileup of library fragments with a UMI and a library-specific multiplex index. So there certainly are other tools, but the majority of our customers follow one of the paths depicted on this diagram. So just to summarize the assay, again, it's a 3-hour turnaround time from DNA to sequenceable library. Really, one thing that is a major differentiator for our product is the very low input. We use sub-nanogram input for microbial genomes and targeted panels, and up to 5 nanograms of input for something human genome size. We are able to capture essentially as long molecules as the customers are able to isolate. So we routinely have molecules in our data set that are greater than 100 kb in length. And our software for de novo assembly and haplotype phasing is freely available. And just to further illustrate the input requirement differences, so if you're using Telseq, again, you're looking at sub-nanogram to 5 nanogram amounts of DNA. And this compares to PacBio and Oxford Nanopore, which need hundreds of nanograms to microgram quantities. And one thing that we've found is we have some customers in the de novo assembly non-model organism space that have been able to successfully use Telseq to be able to prepare genomes from single insects, de novo assemblies of a single individual genome, really getting a very specific genome rather than having to pool multiple samples to prepare a de novo assembly. So now that I've outlined the biochemistry of our library prep and our basic analysis workflow, I'm going to walk through several examples of data. So we're going to start with some microbial examples, then move on to phasing and SV detection and scaffolding, and close out with some targeted phasing data. So like every other molecular biology tool, we're going to start with microbes and work up to larger organisms. If you're not aware, we do work closely with Illumina in many instances, and this is one such example. This is an application note authored by Illumina focused on Telseq for microbial genomes. This application note is available on the Illumina website. It features, it calls out using the MiniSeq and MySeq. That is an Illumina specific marketing thing. Our assay is also, of course, compatible with the NextSeq and NovaSeq as well. Also, this application note is describing the availability of our analysis tools on BaseSpace. So in this application note, we were looking at de novo assembly of a wide range of bacterial genomes in conjunction with Illumina. I want to start with the diagram on the right. So in this study, we looked at a range of GC content. We are able to handle high GC organisms, just as you can see in that diagram. We do see slightly lower library yield since we are a amplicon-based assay, but we can still handle high GC organisms greater than 60% GC content. Just to look at the gel on the left, you may recall from the earlier slide that I said our ideal DNA size is greater than 40 kb. One thing I like about this application note is they were using suboptimal DNA size, and we still see some very good data. So this is an example where they had DNA closer in size to 20 kb, and we're still able to get good assemblies out. So if a customer can't get 40 kb DNA fragments, it's still very likely they can get good data out of Telseq. So there are quite a bit in this diagram, or in this table, so I'm going to walk through it in chunks. The first third of the chart is description of the genomes, of the reference genomes of the organisms surveyed. So you can see that the genome sizes were very standard bacterial genome sizes, really 1.5 to 5.5 megabase in length, so standard representative range. The GC content ranged from 28% to 69% GC. So, a really good range of GC content, pretty representative of the range of biology one would see. The organisms chosen also in some cases contained plasmids, and in one case contained many plasmids and 2 chromosomes. So from low to fairly high complexity individual microbial genomes. Moving to the middle section of the chart, we can see the data that were collected. In this application note, Illumina collected 100x coverage, which we think is usually excessive for a single genome. I believe they collected 100x coverage because that is their standard practice for de novo assembly of bacterial genomes using unlinked reads. For a new de novo assembly project, we recommend starting with 50 to 60x coverage, not 100x coverage. So moving down the table, we can see the SLF size that was calculated by the software. So SLF is an abbreviation for super long DNA fragment. And you can see that our calculated DNA fragment sizes were right around that 20 kb size that we could see on the gel as well going into library prep. Moving down then to the final assembly data, what you can see is the majority of chromosomes and plasmids were able to be assembled into a single contig. So really getting a very contiguous genome assembly with very few misassemblies across the data sets. And we were able to assemble, for most cases, greater than 99% of the reference genome in that single contig assembly. Worst case scenario, we were able to assemble 97% of the genome, and that was one of the highest GC organisms as well. So we were able to still do quite well with challenging base pair content. There's some additional charts and figures in this application note that I'm not going to cover for time purposes, but there's a little more beyond what's presented here if you think this is going to be useful for communicating with your customers. Again, we could assemble into single contigs. Not only were we assembling chromosomes, we were also able to assemble plasmids. So if you have customers that are isolating total DNA from microbes, they should be able to also assemble the contigs with our de novo assembly tools. And so really giving long read caliber data out of an Illumina sequencer. And I do want to qualify that statement by looking at a E. coli genome. So here is just a table comparing the standard E. coli K12 reference strain as sequenced by Telseq, a Oxford nanopore flow cell condition, and standard Illumina unlinked reads. So what we can see right off the bat, Telseq has a great advantage for the DNA input. It was less than a nanogram compared to microgram quantities for both Oxford nanopore and unlinked Illumina. We were able to align the entire genome, whereas Illumina unlinked reads could come nowhere close with their largest alignment, and Oxford nanopore did pretty good, but we were still able to effectively assemble the genome into a single contig, as reflected by the NA50 for the data set. Since we are using the base pair acquisition of Illumina's high quality data, we did not have any misassemblies compared to 2 misassemblies with the Oxford nanopore data. We had less than half of the mismatches per 100 kb than Oxford nanopore dataset, and really nearly no artificially generated indels either, compared to a fairly high rate seen with Oxford nanopore. So again, just for comparing with other platforms, we have very low input, great contiguity of assembly, and very low error rates. So since we are so good at assembling single microbial genomes, we have started to look at assembling microbial communities, so doing a metagenome of an entire sample. What I'm showing you is a preview of a software tool that should be out probably March or April. It is just about finished. So what we would do for analyzing a metagenome sample is very similar to a single microbial genome. We would perform the library prep, sequence the sample, run telread, run tellink, and then we are working to bring a tool online that would assign strain level identity to the assemblies and also quantitate relative abundance. What you see here is a proof of principle study that we've started to present at scientific meetings, where we purchased a 20 species DNA sample from ATCC, where the microbial community members were present at different relative abundances. And what we've found is we were able to identify every species in the sample, and we were able to assemble and assign strain identity to the assemblies of the 3 most abundant bins of microbes. So what we're trying to do at this time is build better guidance about the dynamic range of species we are able to successfully assemble in a metagenomic sample, and build out some more guidance on depth of coverage and best practices. But the software tool is effectively done at this point. So the final software tool will be coming soon. Customers could already start preparing and acquiring the data and doing some of the initial analysis steps today. So I gave you a lot of microbial data. We're going to now pivot to human and non-model organism data. So would like to start with some phasing data with human samples. So most of our human data are going to be from either NA12878 or NA24385, 2 of the genome in a bottle, very highly studied microbial genomes, as you often see from vendor presentations. Looking at the left diagram, you can see both this 12878 and 24385 samples had the vast majority of molecules in the data set were greater than 20 kilobases in length, and a non-trivial amount of the DNA molecules present in the data sets were greater than 100 kb in length. And this becomes important for phasing and a lot of the structural variation observations that we are able to make. Just additionally, in the right-hand chart for the 12878 sample, you can see that we have really good recall rate and precision for both SNPs and indels. Indels obviously being slightly more challenging than SNPs, as it would be for any platform. As I mentioned, our data type are similar to 10x genomics. So I wanted to briefly compare data sets we've been able to generate with some 10x genomics data sets. So with similar depth of coverage, we've observed that we are able to generate phase blocks up to 3 times the length of what has been achievable with 10x genomics, and seeing a roughly similar error rate. So the same quality data, but larger phase blocks. Since the 10x offering is no longer on the market, I don't want to spend too much time on the comparison. I think the important thing to stress when engaging with legacy 10x genomics customers is really the first line on this slide chart, that if they want to use our linked read technology, there is not any capital equipment to purchase that is specific to our assay. So they'll get the similar performance to what they were seeing with 10x genomics with much less upfront investment. Don't want to go through really the rest of the comparison. I want to jump to some data. A lot of the data that I'm going to be showing for the next few slides is from a paper we put out in Genome Research. We'll provide information linking to that paper. It's really a great comprehensive overview of Telseq in much greater detail than I have time to present this morning. What you see here is a representation of phasing of the MHC region, looking at a cluster of class 1 HLA genes and a cluster of class 2 HLA genes. The coloration is the expected haplotype assignment of a given variant across the region. So you should be noticing 2 things. First, that we are able to correctly assign, as expected, the vast majority of the variants. There are a few loci which appear to be misassigned. We go into this a little more in our genome research paper, but the majority of these putative misassignments are in regions of low quality and lower confidence in the reference data set. So a number of these are probably incorrect annotations and not incorrect calls with our software. And we're continuing to investigate some of these regions as well. Additionally, for phasing, here's a great example of where we were able to phase overlapping deletions. So one chromosome had a 19 kb deletion, and the other chromosome had a 114 kb deletion. So quite obviously, if you weren't able to robustly phase the genome, you would probably only be able to catch the 19 kb deletion deleted region, which is missing from both chromosomes. So we can phase diploid genomes, showed you 2 examples of that. And we're able to handle challenging regions that customers will actually care about, such as MHC, which is always popular to be being able to phase those highly polymorphic immune regions of the genome. Before I move away from phase blocks, I did want to get to some data that I mentioned early in my talk. So this is our observed relationship between phase N50 of the phase block and DNA molecule length going into library prep. So What you can see here, this is really one of the data sets that we use to make our recommendation of using DNA greater than 40 kb in length when practical into our data sets. So you can still phase the genome, you can still perform a de novo assembly with DNA fragments shorter than 40 kb in length. The length of the assembled blocks will decrease. So some of this will be project specific as to how big of an assembly is big enough for your customers. So if a customer is concerned they can't isolate 40 kb length DNA, as long as they can isolate DNA greater than 10 kb in length, it's probably worth trying our assay. We can always advise on how to improve contiguity of assembly. The threshold for good enough on an assembly is obviously going to vary project to project. So that's why we have kind of a range of DNA fragment size that our assay can support. I wanted to spend a little bit more time on de novo assembly of larger genomes. And then we'll close out with some targeted phasing and then move on to sales bits after we finish this data discussion. So here is an example of rather than phasing a human genome, we are also able to assemble human genomes with the Telseq assay, and it performs quite well relative to other solutions on the market or that were on the market. So we were able to generate comparably large contigs as 10x genomics, really doing quite well there. We have very few misassemblies comparatively, even for something as complex as a human genome. And comparing all these platforms, we were able to generate the largest alignment and had the largest NA50 of the data sets examined. So really, we expect most people looking at human genomes are going to be more interested in phasing, but if they're interested in de novo assembly, you can use Telseq to prepare libraries for de novo assembly and collect that for human genomes as well. So we can de novo assemble human genomes, really good misassembly rate, and great contig length and N50. Really for de novo, we do expect it's going to be more non-model organisms. And I wanted to share two brief examples of non-model organism genome assemblies that had existing long-read data sets that were honestly pretty good. But even if a customer has a pretty good PacBio assembly or a pretty good Oxford Nanopore assembly, we expect they can further scaffold their assemblies and continue to improve them. Not only is our technology a different biochemistry, so we'd expect just fundamentally different breakpoints in how the data set come together, but we're also able to span greater than 100 kb, which you really can't do with PacBio at all, and you're not going to have very many molecules of that length in an Oxford Nanopore data set either. So if somebody wants to build the best reference genome they will need Telseq, even if they are using existing long-read solutions. I think I hit that. Yeah, so just again, if they want the best genome possible and they already have good data from other long-read products, they can still further refine their genome using Telseq, or they may find once they collect their Telseq data that the de novo assembly is sufficiently good at a much more attractive price point that they would switch to only using Telseq alone for de novo work. I briefly want to speak to two targeted phasing examples. So Telseq was developed as a whole genome focused de novo assembly product, but we've had a lot of interest in the field from people wanting to phase targeted assays. So we have a protocol that works with Agilent SureSelect. That's a protocol that's downloadable from our website. It's a very similar library prep to our standard workflow. We just add in an additional 3 PCR cycles to ensure sufficient product to go into the HIBE. You would then take your library, go into the Agilent SureSelect workflow. It's the nearly identical to the default SureSelect workflow. The only alteration is we provide blocking oligos for purchase that need to be used because we have different sequencing priming sites on our libraries. So we are able to phase exomes and other targeted captures. So for the Agilent SureSelect exome, we phased more than 75% of what was there. You can also use our assay with other HIBE captures. The one thing we've noticed with other platforms, we see a lower on target rate than we do with the Agilent SureSelect workflow. That is likely due to workflow specific optimizations that would need to happen around the blocking oligos. So we're best able to support SureSelect, but we do have other customers trying captures with other platforms. They should just be aware they might see a lower on target rate with our samples if they're not using SureSelect. Just one other, yeah, and so yeah, we work best with SureSelect, but you can use us with most HIBE captures on the market. Just to show one last enrichment story, we've also used Telseq downstream of Sage Bioscience's HLS catch. So this is a CRISPR-Cas9 based enrichment. So using that paired with Telseq, we were able to phase 200 kb regions of the BRCA genes. So we're compatible with multiple enrichment technologies to phase targeted regions if somebody is not interested in whole genome studies. So that was a lot of data. I'm going to now step to talking about kit configuration, and David and I will talk a little bit about our where we've seen sales success and who we see as the ideal customers to reach out to with our current offering. So a new user is going to we would strongly recommend ordering these 4 parts. So our standard kit contains enough reagent for 12 small genomes or 4 human-sized genomes. We sell our library prep across a few boxes. So box 1 contains the enzymes, and box 2 contains our barcoded beads. We sell those as separate boxes because the beads are stored at 4 degrees, the enzymes are stored at -20. So make it a little easier on the customer that they have a visual cue that they need to be stored in different conditions. They will also need library multiplex primers. So if they're just starting out with a small kit, they probably just need 1 set of multiplex primers. And then they also will need the Illumina sequencing primers to spike into their sequencing reaction. So our Illumina sequencing primers do not interfere with the default Illumina libraries. So if a customer wants to run a mixed flow cell of Telseq libraries and standard Illumina libraries, there will not be cross reactivity. Since we do need spiked in sequencing primers. That does mean we are not compatible with Illumina's iSeq instrument, which does not allow for the spike in of sequencing primers. We are compatible with all other Illumina sequencers. We recommend customers start with our standard size kits. Even if they're expecting a larger project, it's useful to get some data in hand just to make sure that our sample coverage assumptions hold true for their project. We find for some mid-sized non-model organisms, really getting that feedback from the data to help estimate how much reagent they would need for a project is useful before they scale up their orders. Here is just some pricing information for those 4 items. Once a customer has tried the low throughput kit, we do offer higher throughput kits as well. So these contain 6 times the volume. So you can process 72 small genomes or 24 human-sized genomes using our high throughput kits. Additionally, we do have 3 sets of multiplex primers. So it's possible to run up to 24 Telseq libraries on a single flow cell. And this hasn't been limiting for customers yet. The only scenario we could think of where 24 samples might be fewer than a customer would want to run would be if it's a very small targeted assay or microbial genomes, but we haven't really encountered that yet. That our multiplex primer sets can come in 3 sets of 8, and we do also have a high throughput version of the Illumina sequencing primer part as well. Just here is some additional pricing information there. I believe you've gotten more detailed price lists than this. Gonna pass it to David briefly to talk about sales and marketing support.
Yes, indeed. Thank you, everyone. David Nordell with Universal Sequencing Technology. I am the inside sales manager here. I'm your primary point of contact for all of your purchase orders and any help with quotes or anything like that. We do have a lead sharing program. I'm not sure if Dr. Levy has sent that first batch of leads over to you. I'm sure if he hasn't, I'm sure he'll be doing that within the next.
I've done the first one.
Okay, perfect.
For my call.
Yeah.
So we'll talk to Anna as well.
Perfect. Yeah. So we do, on a regular basis, do some lead gathering for you on your behalf with our lead provider, and then we distribute those, as needed. We offer a weekly Q&A with our scientific and technical team, including Andrew, who's on the call today, and then Dr. Tom Chen as well. And that is every Thursday at 8 a.m. Pacific time. So that would be 5 p.m. Central European time. And that's typically the best opportunity for customers and others to come and ask questions, anything that they may have application specific or general questions. We encourage them to come and join us at that Q&A. We do work with you guys on marketing material and generating press releases. So that's something that Dr. Levy and I will be working with your team on. And placing orders is pretty simple. You can just go to universalsequencing.shop. You select the items right there, and then we do actually have customer accounts as well that you can set up so that your billing and shipping information is right in there, and then you just upload your purchase order in that screen. It doesn't take any payment right then, and it then goes into our system for processing. Alternatively, you can also email your purchase orders to sales@universalsequencing.com. Just kind of depends on what you want to do. We do find that the website tends to be fairly easy for people to use when, we're unavailable or it's outside business hours. So sometimes with the different geographies that we're working with, that does tend to be an advantage. So if you have any questions on placing an order or anything like that, feel free to reach out to me. You can reach me by phone or email, and I'm happy to answer any questions. Thank you.
All right, and just wanted to go through a few more things before opening it up for Q&A. Just at a high level, we've seen our early customers fall into one of three categories. So really a lot of interest in the non-model organism reference genome space. These types of users generally are quick to bring on new technologies to further improve their projects. So we have seen good interest across a wide number of organism types. I should mention, because I don't think I called it out too specifically, our software has been extensively tested on diploid organisms. We're still trying to build more guidance for polyploid organisms. So if a customer is looking at polyploid plant genomes, they may need to perform some other type of analysis outside of our software package. We're still trying to understand how our software performs with polyploid genomes. So I'd like to make that clear. I don't think I mentioned that earlier in the talk. So additionally, obviously, we have that great app note out with Illumina. So we are great at microbial identification and characterization. And really the third set of customers that we're seeing a lot of interest with are people that had used 10x Genomics linked read solution, really liked that solution, and are looking to close out projects or start new projects with that same type of technology. And as we continue to grow our customer base, we're already a global company. As we continue to grow our distributor network with folks such as yourself, we expect this map to get further filled in. But we do have customers the world over, so lots of different projects, lots of different geographies, lots of fun science. And as we're building up our customer base, we expect the list of papers and customer success stories to grow. But here are some preliminary resources for your technical discussions with customers. So you're probably aware of our corporate website. I'm sure most of you already looked at that. So that's where you can find our protocols, our software, and that will continue to grow. We do have a video on YouTube of a cartoon animation of how our assay works. Then there are a few webinars with customer talks. So we have from the Earth Biogenome Project, there's a talk hosted on a server at the University of Illinois. It's from a fairly extensive symposium. There are two talks featuring Telseq prominently at those timestamps. There was also a Genome Web webinar that was actually sponsored by Illumina, but features exclusively Telseq data in a insect de novo assembly story from the same group at University of Illinois. As I mentioned in my talk, we have that paper in genome research that is really the most comprehensive Telseq publication to date. And the application note for the microbial de novo sequencing is freely available from the Illumina corporate website. I would just like to mention that most of the data I showed you was from collaborations with one or more groups. We collaborate very deeply with Illumina. There was also some data we generated in collaboration with SAGE, and most of the non-model organism data was from Alvaro Hernandez at the University of Illinois. So we really appreciate all of the work they've done with us. Just to close out again with the summary, Telseq is a simple one-tube, fast and easy linked library kit. Basically converts a Illumina sequencer into a highly accurate long-read sequencer data type. And our assay requires very low DNA input, so really great for precious samples or material that's difficult to obtain a lot of. So just as a reminder, you can route your technical questions to me, sales questions to David, and like to thank you for your attention and open it up to questions.
Before we start, Joanna and Michael, first of all, thank you very much, Andrew and David. And it's good to see you, Michael, again. And Joanna, nice to meet you. I have a call with your friend, Michael, in a few minutes. You know who I'm talking about. So I'll take that and I'll click off from here. Please continue with the Q&A and looking forward to working with you this year. Thank you.
Thank you, Aaron.
Great. Thank you very much. Let's see if we have any questions in the audience. Someone raised a hand. I can't see now who. Please.
Maybe it was me. Can you hear me?
Yes.
Yes.
Oh, great. I was wondering if any of your customers has used this technology together with bisulfite sequencing, or to look for methylation, essentially.
So we haven't to date. I can check with, I'll make a note to check with our CSO. I think the challenge, I'll check for sure, but I think the challenge would be the initial bisulfide treatment might damage the DNA such that you'd lose some of the positional information. So I'm not sure at I'm not sure on compatibility with bisulfite. I'll confirm and get back with more detail.
Thank you. Maybe I can ask another question.
Sure.
Is there any bias in terms of sequencing? Like, do you see, in terms of like repetitive sequences, or, segments that are usually folded?
So there doesn't seem to be anything that's specifically biased. We will see, since it is transposon based, you will see certain regions of lower coverage it tends to be organism and data set dependent. There isn't any sort of known motif that's difficult for us per se. I'd say the one scenario for repetitive motifs would be if the repeat is a fairly large repeat, such that so our library inserts are typically 150 to 200 base pair. If it was something that just happened to be a repetitive element of that size, that still would be challenging for us. So if we had a 200 base pair repetitive element with a 200 base pair library insert, that would still be challenging to deconvolve. But that would really be the only systematic weakness that I'm aware of.
Thank you.
Yeah, that was actually my question too. I have another question. You kind of indicated that in your talk, but I guess the technology that comes closest to yours, that's Loop Genomics or Element Biosciences. They were just acquired, I understand. So how do you position yourself against them when you talk to customers?
Sure. So Loop's the currently on market assays for Loop are really focused on 16S enrichment specifically. So we tend to not compete with those customers. What we're bringing online first for metagenomics is the full genome assembly. So if they care more about more than just 16S and are looking at microbial communities, we would be the superior solution. If they only care about 16S, honestly, today we don't have a great on-market solution. We are working on, so our next iteration in the metagenomics space will be a read-specific product that's not assembly-specific. That might be more amenable to that space. It's slightly different use cases as well. And the other similar product, the other linked read product on the market, or that's coming to market, is going to be a Illumina internal product, and that really is still shorter read lengths than we're looking at. So with the Illumina, I believe it's Infinity product that had been announced, that's really only fragment sizes up to 10 KB. So we're really operating in a exceptionally long range assembly size. So most of the other products on market are really hitting that size range that would be where PacBio is hitting. We're going beyond that with much longer molecules.
Andrew, thank you very much for this very, very interesting presentation and really complete. I think you can make it 3 hours without problem. But you presented very good examples on HLA. Did one of your customer ever used it for TCR repertoire sequencing of immune cells, which are somatic gene rearrangements and mutations?
So I don't offhand have any TCR specific data sets. We do have, I believe we have one of the 12.8, I think we have the 12.878 and 24385 data sets available that we could share if somebody wanted to just see what our data look like for those regions. That's something that I know customers are quite interested in, probably right up there with MHC, right, for phasing.
Yeah.
So we will have exciting data sets. I don't know that I have anything other than 12.878 and 24385 available currently. And those are going to be a little more whole genome focused rather than targeted, of course.
Yeah. And if anything come to mind, can you share it with us, please? Because
Sure.
genomics is really coming to the edge now. So thank you.
And that's where we expect most of the human phasing interest to be, of course.
Thank you.
Sorry, maybe I can ask 2 more questions.
Sure.
That's all right. So you mentioned the raw data that you get out of this.
Mm-hmm.
So is it possible for the end user to use that raw data and do their own assembly? Or
certainly, yeah. So most of our users are using our tools, but after, and here I'll go back to the software workflow diagram. So the customers will always have to run our denoising and QC tool. So that Telread tool at kind of the top. The output from Telread is a QC HTML file, and then the FASTQ data of the cleaned up reads. So the data will be an 18 base pair UMI for the index 1, and index 2 is the 8 base pair multiplex primer index, and then you just have your 200 base pairs of sequence in the middle. So if customer wants to take that library fragment type and do something else with it, they certainly can. The software makes the FASTQs available, mid-process. Each of the 3 tools we provide is typically run on its own discreetly and has an output you can manipulate.
Okay, so it's not impossible for the user to link the reads by themselves, if I understand you correctly.
Right. Shouldn't be. It would just depend. We'd expect that would be kind of a very informatic savvy customer. But if they didn't like our tools, didn't want to use 10x genomics tools, there are other tools out there, certainly. We expect most people to want either our tools or 10x tools, but at least based on our interactions with customers to date.
Right. Thank you. And then for my second question is, could one use this for single cell sequencing, or is that not sensitive enough?
So our current workflow is not going to be directly for single cell. I'm not sure how much we're saying beyond generalities. We are working towards something specific for single cell. There will be a hardware component for that too, though, and I don't have a release date. So the assumption is we would have something better for single cell than this. I don't know how much has been done with our reagent alone outside of other instrumentation pairings for single cell, if that makes sense.
Thanks.
For whole genome sequencing, would it be possible to kind of replace the classical karyotype assays, by this kind of sequencing methods, approaches?
So you should be able to detect rearrangements, copy number changes.
Yeah.
I think that would come
because you can assemble a whole genome. So can you position it as well on the chromosomes?
Yeah, so we so we are able to detect structural variations. You should be able to detect breakpoints. I guess the question would be sort of individual lab need in that space, but the great thing about long read sequencing, whether it's linked reads, true long reads, any of that, the longer reads should be able to really be much more robust at breakpoint detection than short reads. So I think I would say most of the application you're asking for should be manageable with our linked reads. There might be, depending on specific use case wants, but at a high level, yes.
But you have no example to share of a user having applied it, to your knowledge.
So not in a specific sense. The best example of what we can do with structural variation detection for human genomes would be currently is in our genome research paper.
Okay.
So that's where the MHC data is. That's where there are a bunch of additional examples I didn't have time to go through of deletions, fusions, et cetera. So kind of gives a more comprehensive overview of variant types we can detect in that paper.
Thank you.
So, any more questions? Yep.
I can ask my last one, actually.
Please.
Which is, are there any plans to sort of transfer this idea technology to sequence long RNA? I know long RNA would be single-stranded, so you probably need to convert it to double-stranded, but.
So We are going to very soon have an application note focus describing amplicon sequencing. So I think for RNA, you'd have to generate cDNA. I think one of the things we're still trying to determine in the amplicon space is what the shortest fragments we can really add value and be able to reliably link. I know for sure we can go down to like 3.5 KB fragments. I forget what else we're looking at in terms of proof of concept for lower limits. So the minor concern for RNA studies would be, I'm not sure how much of the cDNA pool we'd be able to link. We're still trying to appreciate what the lower limit of DNA fragment size we can reliably link is. If that makes sense.
Would it help to use some sort of selects method where, because you mentioned amplicon sequencing, but in this way, you select the ones, amplify those, and re-amplify those again, so you get cycles of amplification on your mRNA of interest here.
Potentially. So I just know from what our R&D is doing, really in the shorter fragment space, we're looking at amplicons. I don't think we are currently doing. I don't think we've done much like RNA specific. We've really been focused much more on the DNA side and on really the larger fragment side. Long term, I don't know what our plans are for RNA. Short term, really, I think we have confirmed the best fit with high molecular weight DNA projects.
Super. I guess these were the technical questions.